Hello. Apakah? Apa salah aku stop presenting lah. Sekejap. Okay, so hari ni kita akan sambung topik 6, gene regulation and expression like operon. Okay, so these are the learning objective that we're going to discuss for today. The objectives are explain the concept of operon and gene regulation, state the components of operon, explain the components of like operon and their function in E. coli, explain the mechanism of the operon in the absence and presence of lactose. Lecture dah habis ke belum? Ni. Lecture dah habis ke topik 6? Anyone? Lecture dah habis ke topik 6? Aku tak dengar. Lecture dah habis ke topik 6? Boleh tulis tak kat message tu? Aku love. Kenapa? Baru tadi kelas habis ke 12.30. Okey je. Salah laptop ni. Dah habis ke belum? Topik 6. Lecture. Belum. Okay, at last thank you. Ada juga yang bagi respon. Okay. Okay. Kalau belum, mestinya awak tak discuss lagi lah yang ni. So, sebelum kita discuss apa itu lag operon, saya nak bagi tahu cara tulis lag operon. Lag kena underline. Okay, lag tu you must underline lag. Kalau awak tak nak underline, awak italic kan. So, if you type, okay, if you use computer, make sure you underline or you italic. But if you use handwriting, okay, you write by hand, the word lag you must underline. You cannot italic. Okay, sebab kalau tulisan awak memang sangat, macam mana kita nak tahu itu adalah italic? Okay. Explain the concept of operon and gene regulation. State the components of operon. Explain the components of lag operon and their function in E. coli. E. coli ni, okay, E. coli ni awak cara tulis E. coli kena tulis macam ni. Underline E, underline coli. Okay, underline E, underline coli. Kena dua garis yang berbeza. Tak boleh sekali kan. Okay, so first kita tengok apa itu operon. The mechanism for this control of gene expression in bacteria is described as the operon model. Okay, operon is referred to a control of a cluster of genes as a single unit. Okay, and operon only exists in bacteria. Okay, operon is only exists in bacteria. So the one that we learn in our syllabus is lab operon in E. coli. Okay, lab operon in E. coli. The importance of operon is enable the bacteria to adjust their metabolism to environmental change. Okay, and enables the bacteria to adjust their metabolism to environmental change. So this is example of operon model or the lag operon. The lag operon in E. coli uses 3 enzymes. Okay, it uses 3 enzymes to absorb or take up and metabolize lactose. So the 3 enzyme is coded by lag Z, lag Y or lag A. Lag Z, lag Y and lag A. Okay, the 3 enzyme tu codes by lag Z, lag Y, lag A. Lag Z, lag Y, lag A is also known as structural genes. Okay, structural genes. So the lag operon is started from promoter until lag A. Okay, kuatlah pula. Sekejap, slow kan. Okay, lag operon ni dia punya komponen starting from promoter sampailah kepada lag A. Lag I or regulatory gene is not included in the lag operon. Okay, so each of these you need to know the functions. Okay, you need to know the function for each component in lag operon. So the promoter promoter, the function it act as binding site. It act as binding site for RNA polymerase. Operator, binding site for repressor protein. It also act as switch on and switch off for the operon. Operator ni macam switch juga. Dia akan switch on ataupun switch offkan the operon. It also have three structural genes. The three structural genes are leg Z, leg Y, and leg A. And each gene codes for each enzymes. For example, leg Z codes for beta galactosidase, leg Y codes for permease, leg A codes for transacetylase. Because this is genes, you cannot write produce. Awak tak boleh tulis like Z produce, like Z synthesize. Kalau dia gene, dia kena codes. Okay, codes for beta galactosidase, permease, transacetylase. Cara tulis like Z, like Y, like A macam ni lah. Like dengan huruf tu kena rapatkan. Okay, like Y rapatkan, like A rapatkan. Okay. Other than that, you need to know about the functions of each enzymes. Okay, the function for each enzyme. Beta galactosidase, permease, and transacetylase. So what is the function of beta galactosidase? Beta galactosidase, it will hydrolyze lactose to glucose and galactose. Okay, that is the function of beta galactosidase. Hydrolyze lactose to glucose and galactose. So maksudnya, kalau awak tahu, awak memang tahulah lactose ni apa dia? Lactose ni apa? Kita ada karbohidrat, kita ada 3 classes kan? Lactose ni di grup mana?
Disaccharide.
Grup mana? Lactose.
Disaccharide.
Disaccharide, betul. Sakinah cakap disaccharide. So this lactose disaccharide dia akan tukar kepada inilah monosaccharide. Monosaccharide. So this lactose will be hydrolyzed into glucose and also galactose. Okay. We also have permease. Permease, the function is something to do with permeability. Sebab itulah nama dia permease, permeability. So permease, it will increase the permeability of the membrane protein that transport lactose into the cell. So as you all know, membrane, kita punya plasma membrane is made up from phospholipid bilayer. So this enzyme, this permease enzyme will increase the permeability of membrane protein so that more lactose will transport into the cell. Okay, that is permease. Okay, then we have transacetylase. Trans ni maksudnya transfer. Acetylase adalah acetyl group. So the function is to transfer acetyl group from acetyl-CoA to beta galactosides. Okay, itu adalah function untuk this enzyme. Okay, function untuk transacetylase. Ada 2 function. First, transfer acetyl group from acetyl-CoA to beta-galactoside. Second, it will detoxify other molecules entering the cell via permease. Okay. Itu adalah kita punya leg operon. Kalau awak tengok kat sini, kita ada leg I. Leg I is not included in the leg operon. Leg I is also known as regulatory gene. The regulatory gene, the function of regulatory gene is to it will code for repressor protein. Okay, regulatory gene or leg I located outside the operon, it will code for repressor protein. Okay, it will code for repressor protein. Okay, yang ni lah. Leg I ni, regulatory gene, dia akan code for repressor protein. Maksudnya gene ni dia akan undergo transcription dulu. Okay, it will undergo transcription to produce mRNA, and this mRNA will undergo translation to produce protein. And that protein is known as repressor protein. Okay. So this is the promoter. The promoter is whereby the RNA polymerase will bind to it. The operator is whereby the repressor protein will bind to it. The operator tempat repressor protein bind and also act as switch on or switch off. These are the structural genes, but it cannot seen here. Only we only can see like Z. In this diagram, we only can see like Z. Okay, so like Z, like Y, like A. Kalau tak ada transcription of like Z, like Y, like A, so no RNA made. Bila tak ada RNA, tak ada protein, tak adalah enzymes. Okay, so I will explain how lack operon regulates. Okay, how lack operon regulates in the absence of lactose. Maksudnya tak ada lactose. Tak ada lactose, lack operon akan switch off. Okay, lack operon, no lactose, it will be switch off. How? Okay, macam mana dia boleh So macam mana dia boleh switch off? First, we have a regulatory gene. Okay, macam mana dia boleh switch off kalau tak ada lactose? Okay, tak ada lactose. Maksudnya kita tak minum. Maksudnya tak ada lactose lah. Tak ada dalam badan tu ada, dalam environment tu tak ada lactose. So bila tak ada lactose, dia akan switch off. Kenapa boleh switch off? Okay, maksudnya once it switch off, repressor protein is active. Okay, this repressor protein is active. When this repressor protein is active, it will bind to the operator. Okay, it will bind to the operator. Once it bind to the operator, okay, RNA polymerase cannot bind to the promoter. Okay, RNA polymerase cannot bind to promoter. So when RNA polymerase cannot bind to the promoter, okay, there is no transcription of structural genes. Okay, no transcription of structural genes. So bila tak ada transcription, tak adalah mRNA, tak adalah protein, tak adalah enzyme. That's why dia adalah switch off. Okay, itu switch off. Sebab apa dia switch off? Sebab protein ni aktif. Repressor protein is active. It will bind to the operator. Okay, so that is in the absence of lactose. In the presence of lactose, lactose ada, dia akan switch on. Okay, the lac operon will switch on. So what happen is this lac I, okay, lac I will code for mRNA. mRNA will undergo translation, will produce protein. So bila ada lactose, this lactose will converted to its isomer, which is allolactose. Okay, lactose will converted to its isomer, allolactose. Kalau hijau ni adalah allolactose. Okay, yang kalau hijau ni adalah allolactose. And this allolactose will bind to the protein. Okay, allolactose will bind to the protein. And this will make the repressor protein will become inactive. Bila inactive, macam mana dia boleh jadi inactive? Tengok. Daripada bentuk dia macam ni kepada bentuk macam ni. Bentuk dia lain. So bila bentuk dia lain, dia dah tak boleh bind. Okay, dia dah tak complementary. So dia tak boleh bind. So the repressor protein is inactive, so it cannot bind to the operator. So when it cannot bind to the operator, where did the allolactose come from? Dia tak come from apa-apa. Dia akan convert. Lactose ni, dia punya isomer adalah allolactose. So bila kita dah ada lactose dalam badan, some of the lactose will be converted to its isomer, which is allolactose. And that allolactose will act as inducer. It will act as inducer. And it will bind to the repressor protein. Okay, it will bind to the repressor protein, making the repressor protein become inactive. So when it is inactive, the repressor protein cannot bind to the operator. So when it cannot bind to the operator, RNA polymerase will bind to the promoter and it will transcribe like Z, like Y, and like A. And mRNA will be produced. That mRNA, welcome, that mRNA will codes for like Z, mRNA will codes for beta galactosidase, like Y codes for permease, like A for transacetylase. Dapat je beta galactosidase buat apa? Dia akan hydrolysekan. Okay, it will hydrolyze lactose to become glucose and galactose. So dapatlah dia energy. Okay. Alright. So ni adalah gambaran secara GIF. So kita ada promoter, binding site for RNA polymerase. Okay, that is inducer, allolactose. It will bind to the repressor protein, change the shape. Repressor protein is inactive. So it cannot bind to the operator. So when it cannot bind to the operator, RNA polymerase, it will bind to the promoter and then it will transcribe like Z, like Y, like A genes. And then the three enzyme will be synthesized. Right? So dapatlah yang tu. Regulatory gene tak faham sangat. Regulatory gene ni, regulatory gene ni adalah leg I. Dia adalah leg I. Okay, dia adalah leg I. Leg I ni, dia gene lah kan. So this leg I, ni, leg I ni dia akan synthesize, dia akan undergo transcription to produce mRNA. And that mRNA okay, undergo translation to produce protein. Basically, leg I ni, this leg I or this regulatory gene codes for repressor protein. Okay, leg I codes for repressor protein. Yang tu. Boleh ke? Lanjut, boleh ke? Okay, leg I codes for repressor protein. Yang tu. Okay, sama je. Leg I ni adalah regulatory gene. Ni, leg I adalah regulatory gene. Codes for repressor protein. Repressor protein adalah yang menentukan operon tu switch on or switch off. Okay, boleh ke? Siapa boleh? Okay, thank you. Alright, boleh setakat ni? Angkat tangan kalau siapa boleh. Okay, thank you. Susah, tak ada suara. Okay, kejap. Alright, kalau boleh. Sekejap Before we proceed to chapter 7, okay, before I proceed to chapter 7, I want you to answer, wait. Sekejap Okay. Sebentar. Cekeman. Okay, kita buat quizzes. Okay, kita buat quizzes, masukkan kod 411930. Saya akan tanya lab operon. 10 soalan sahaja. Okay, 10 soalan sahaja. Right? So, join lah. Bolehlah join sekarang. Okay, lagi 6 orang. Baru 22. Lagi 3 orang. Lagi siapa? Kita tak nampak. Oh, sorry. Tak nampak. Okay, so 27 lah. Lagi 2 orang. Lagi 2 orang. Anis ada ke? Anis Nurjiha. Anis ada ke? Anis ada. Ni ada je. Ada je dalam ni. Huh? Crazy. Yeah. Hello, habis dah. Ya Allah. Okay. So, sekejap First Anas, second siapa tadi? Second Manisha, third Keying. Okay. Soalan apa ni? Question 3. Question 3 What is 1 in the diagram? Ada mana 1? Dia tunjuk tak 1? 1 is in diagram is RNA polymerase. Okay. Okay, question number 5. Okay, question number 5. Functioning of unit DNA containing cluster of genes under the control of single regulatory signal or promoter is termed as operon. So yang ni lah operon. Mana ada? Dia dah cakap cluster of genes. So bila cluster of genes, dia adalah refer kepada operon. Just now the one cannot be seen in the first question. Apa dia cannot be seen? First question, this one Ni ke? Ini ke? Okay, Tina. Which question? Sebab question dia jumbled up. Question number 3. This one. What is 1 in the diagram? 1. Ni, kalau kuning ni. Oh, saya tahu kenapa tak boleh nampak. Sebab dark kan? Sebab you jawab quizzes tu, dia macam dark mode macam tu. Okay, tak apa. Okay, so next time I tahulah, I tak buat lah. I tak masukkan soalan macam ni. Okay, so tak apa. Alright, next question. Question 6. Kita tengok question 6. Which of the following condition leads to maximal expression of the lactoferrin? Lactose present, glucose absence. Okay, so kalau lactose present, glucose absent, barulah dia akan ada maximal expression of the lactoferrin. Maksudnya lactoferrin tu akan bekerja secara maksimal. Kalau dah ada glucose, lactoferrin tak ada dah. Dia tak perlu buat. Dia akan switch off. Jadi kalau ada glucose, dia akan switch off. Tapi kalau ada lactose, tapi tak ada glucose, barulah dia akan switch on. And dia akan bekerja secara maksimum. Okay. Alright. So that's all. Okay. Ada soalan ke? Nak tanya? Soalan ke? Kalau tak ada soalan, saya nak sambung chapter 7. Okay, saya nak sambung chapter 7. Sikit je. Saya tak adalah cerita sampai gene mutation sampai habis. Mungkin saya cerita pasal sampai apa sampai base substitution saja. Okay, sekejap Batuk sekejap. Batuk. Chapter 7. Which one is chapter 7? This one. Okay, chapter 7. Alright. For chapter 7, okay, chapter 7 mutation. Okay, this is mutation. Chapter 7, topic 7, mutation. So ini gambar apa ni? Mutation disebabkan apa ni? Apa nama condition ni? Kita panggil sebagai apa?
Down syndrome.
Okay, betul. Okay, Jude cakap Down syndrome. Yes, betul. Down syndrome. Sakinah pun cakap benda yang sama, Down syndrome. Tapi Sakinah punya ejaan betul lah capital D. Okay, capital D. D tu kena huruf besar. Sebab dia adalah term. Kalau term kena betul. Okay. Alright. So for this topic, we have 3 subtopics. 7.1, mutation, classification and types, gene mutation, chromosomal mutations. So for today, we only going to discuss until 7.2. 7.2 ni pun tak habis pun kita discuss. Sikit je kita discuss. So 7.1 mutation classification and types. I'm going to explain everything. Explain mutation, classify mutation into two gene endpoint mutation, chromosomal mutation, state two types of mutation, spontaneous mutation, induced mutation, define mutation, state types of mutation, physical and chemical. Okay, physical and chemical. All right. So first you need to know about mutation, definition of mutation. Okay, definition is a sudden permanent change in the amount, arrangement or structure of the DNA of an organism. The one that I bold, okay, the one that I bold is the keyword. Okay, a sudden permanent change in the amount, arrangement or structure in the DNA of an organism. Okay, ataupun okay, sudden permanent change in gene or chromosome in organism. So there are two definition you can remember mana-mana je lah. Okay. So the mutation, it can be passed to the next generation. Alright, and we have two classification of mutation. We have gene point mutation and we also have chromosomal mutation. A gene point mutation kita ada 4. Kita tengok satu-satu. Chromosomal mutation we have 2. We have structural mutation, okay, which can be described as chromosomal aberration. Aberration ni maksudnya structure. Kita ubah dia punya structure. Okay, mutation, apabila berlakunya mutation, dia akan ubah dia punya structure. Yang kedua adalah alteration of chromosome number. Okay, yang banyaknya nanti kita akan tengok kat sini lah. Kita akan jumpa Klinefelter, Turner, Cry-Ducat, apa lagi? Cry-Ducat sini. Apa lagi? Down syndrome, semua kita akan jumpa kat sini. Okay. So mutation, we have two apa ni? We have two types mutation. We have spontaneous and induced. Spontaneous is occur naturally. Okay, occur naturally without human interventions. So this spontaneous mutation is due to errors during DNA replication. So masa kita tengah masa sel kita tengah buat masa replicate DNA tu, mungkin sepatutnya kita buat, kita tambah base A patutnya complementary to T. So mungkin dia tambah base A patut dengan T kan. Patutnya dia tambah T. Mungkin dia akan tambah, dia silap tambah G for example. Okay. So error yang macam tu lah akan menyebabkan berlakunya mutation. And that is spontaneous. Bukan kita nak. Okay, bukan kita nak. Dia secara naturally. Induced mutation is organism is artificially exposed to mutation. Kita sendiri yang penyebab. Okay, penyebab yang berlaku mutation. Kita sendiri exposekan diri kita dengan mutation. So we have two. We have physical mutation and chemical mutation. Okay, an induced mutation will contribute to genetic variation or genetic evolution at higher rate. Okay, itu adalah induced mutation. It will contribute to genetic variation or evolution at higher rate. Alright. Okay, mutation. Mutation adalah agent that induce changes in DNA and cause mutation. Okay, agent yang menyebabkan dalam DNA kita berubah and berlakunya mutation. Itu adalah mutation. And mutation we have two types. Okay, we have physical and chemical. Physical is high energy radiation such as X-ray, UV ray, alpha ray, beta ray, gamma ray. Okay, ada banyak. Kita awak ingat X-ray, UV ray cukup. If that is physical agent. Chemical agent is colchicine, ethidium bromide, proflavin, mustard gas, nitrous acid, formaldehyde. Okay, ini adalah chemical agent lah. Okay, banyak kita ingat satu dua cukup. Selalunya saya ingat colchicine. Colchicine ni awak jumpa lagi dalam 7.3 nanti. Okay, chromosomal mutation. So sebab colchicine ni, dia adalah agent, it will inhibit spindle fiber formation. Okay, it will inhibit spindle fiber formation. That is colchicine. Okay, and the physical agent here, okay, the X-ray, UV ray. UV ray ni ada lah, macam kita keluar pun ada je UV ray. Tapi dia tak adalah bagi harmful effect sangat kepada badan. X-ray ni memang bahayalah. Okay, dia ada high energy yang sebab itulah kalau siapa-siapa yang nak pergi X-ray, dia akan tanya dulu, awak kecil lagi tak kot? Contoh yang dah besar-besar, okay, perempuan especially yang dah besar tu dia akan tanya, macam saya contoh dia akan tanya, pregnant ke? Contoh, dia akan tanya macam tu. Sebab kalau pregnant tak boleh masuk lah bilik X-ray tu. Okay, sebab dia akan menyebabkan mutation. Okay, mutation kepada fetus. Alright, so that is mutation. Okay, selalu dia tanya, dia suruh bagi 2 ataupun 1 example saja. Okay, mutant. Mutant is organism carrying a gene that has undergone a mutation. So for example that ni lah example yang senang. Teenage Mutant Ninja Turtle. Okay, Teenage Mutant Ninja Turtle, dia punya gene tu dah undergo mutation. So organism that carrying a gene that has undergone a mutation, we call that as mutant. Okay, we call that as mutant. This is mutant. Okay, mutant. Teenage Mutant Ninja Turtle. Okay, it will shows new phenotype as a result from mutation. Okay, dia akan menunjukkan phenotype yang baru. Okay, sebab phenotype yang baru itulah hasilnya daripada mutation. Okay, so that is 7.1. Okay, 7.1. Ada soalan ke setakat ni? Soalan ke? Tak ada. Okay. Alright. So fahamlah baru 7.1. 7.2, gene mutation. Explain gene mutation. Classify the 4 types of gene mutation: base substitution, base insertion, base deletion, base inversion. Describe base substitution, example sickle cell anemia, as missense mutation. Explain frame shift, explain base insertion as a frame shift mutation, explain deletion as frame shift mutation. Okay, so apa yang saya nak discuss mungkin sampai sini saja. Okay, sampai base substitution saja. Lepas tu habis. Alright? Okay. Gene mutation. Apa itu gene mutation? Apa maksud dia? A change occur in the sequence of base of nucleotide base pair in a single gene of a DNA. Perubahan yang berlaku dekat sequence of nucleotide. Okay, sequence of nucleotide in a single gene of DNA. So kita ada DNA, so perubahan yang berlaku dekat situ. Okay, mutation yang berlaku dekat situ. Sebab tu kita panggil sebagai gene. Gene mutation. Sebab dia spesifik kepada dekat nucleotide. If the change involve one nucleotide pair, then it is called as point mutation. Okay, then it is called as point mutation. Kalau melibatkan satu pair saja, kita panggil sebagai point mutation. So bila DNA berubah, amino acid akan berubah lah. Protein akan berubah lah. So different protein may not function as normal. Okay, as a result, different protein patutnya protein tu dia buat function macam ni. Macam dia buat function untuk tukarkan A kepada B. Tapi bila hasilkan protein yang lain, jadilah lain. Okay, dia tak boleh function lah sebagai A tukar kepada B tu. Okay, dia ada lain. So gene mutation kita boleh classify into 4. Kita ada base substitution, base insertion, base deletion, base inversion. Untuk gene mutation ni wajib tulis perkataan base. Okay, I repeat, in exam or in apa-apa je lah, wajib tulis perkataan base. Okay, base ni wajib tulis, tak tulis salah. Okay, salah. Tak ada markah bonus ke, tak ada. Okay, wajib tulis. Untuk gene mutation wajib tulis base. Base substitution, base insertion, base deletion, base inversion. So base insertion and base deletion, it will cause frame shift. Okay, it will cause frame shift mutation. Nanti kita tengok. Okay, apa itu base substitution? Base substitution apabila one or more nucleotide pair replace. Keyword dia sama. Keyword dia sama. Keyword untuk definition, untuk setiap base, definition untuk setiap satu ni semua sama. Tapi yang tukarnya adalah sini, replace. Kalau insertion, insert. Kalau deletion, delete, remove or delete. Kalau inversion, inverse. Itulah. Ni yang lain semua sama. Ni yang almost sama ataupun sama. Kalau ayat depan ayat belakang sama. So kita tengok apa itu base substitution. Base substitution apabila one or more nucleotide pair replaced by another. Dia digantikan. It replaced by another. Digantikan dengan sequence of DNA nucleotide of a chain. So if one nucleotide pair is replaced, kita dah replace dekat DNA. Dekat DNA tu kita dah tukar. So apa yang jadi, mRNA pun akan tukar. Okay, if one nucleotide pair is replaced by another nucleotide pair, then gene akan transcribe kepada different mRNA. Different mRNA akan membawa maksud different polypeptide. Contohnya, yang inilah base substitution. Ini yang wild type, ini yang before mutation. Ini before. So before mutation kita ada GTG, TTC, ATA. Yang ni, ATA. GTG, ATT. Ni DNA. mRNA dia CAC, AAG, UAU. Sini U. Betul lah. Kalau A punya DNA, mRNA dia akan complementary dengan U. So, bila kita transcribe jadi amino acid, CAC akan bermaksud histine, AAG lysine, UAU tyrosine, histine. Katalah berlakunya mutation, base substitution. A tadi tu kita gantikan dengan C. So for example, this A base we replace with C. Okay, we replace with C. Nampak tak C tu? Nampak ke? Inilah. A tu digantikan dengan C. A is replaced with C. C, dia punya partner, kalau DNA dah C, dia punya partner untuk mRNA akan jadi G. So tu maksudnya nucleotide base pair. Kita dah gantikan pasangan dia juga, bukan ganti dia seorang je. Kita dah gantikan kedua-dua pasangan ni. Okay, satu pasangan ni. Partner dia juga kita ganti. So C akan complementary to G. So bila kita translate mRNA menjadi amino acid, CAC-AAG. CAC histine, AAG lysine. UAG ni stop codon. Okay, UAG adalah stop codon. So apa yang akan jadi, kita akan dapat protein yang ada 2 amino acid saja. Okay. Lain yang macam ni, kita akan dapat protein yang ada 4 amino acid. Patutnya kita dapat macam ni, tapi kita dapat macam ni. Itu maksudnya base substitution. Okay. And base substitution ni kita ada 3 jenis. Kita ada 3 types. Okay, kita ada silent. Okay, we have silent, we have missense and nonsense. Okay, silent mutation, senyap, missense, nonsense mutation. Okay, apa itu silent mutation? Silent mutation if the change in nucleotide pair transform one codon into another that is translated into the same amino acid. Kita dah tukar pair dia tu. Okay, contoh kat sini kita dah tukar. Okay, kita dah tukar pair. Tapi dia still membawa maksud amino acid yang sama. Okay, silent mutation ni kita dah tukar nucleotide pair, tukar codon, tapi still bagi maksud amino acid yang sama. So itu tak ada effect. Tak ada effect on phenotype. That is silent mutation. Contohnya yang ini. Okay, kita tengok mRNA before mutation, kita ada UAU. UAU maksud dia tyrosine. Kita tengok silent mutation. Silent mutation, U ni digantikan dengan C. Jadi UAC. UAC ni sama juga membawa maksud tyrosine. Mutation berlaku. Tapi dia still membawa maksud same amino acid. So kita panggil sebagai silent mutation. Mutation berlaku tapi same amino acid, silent mutation. Okay. Missense. Missense adalah change in nucleotide pair transform one codon into another that is translate into different amino acid. Itu adalah missense. Kita tukar codon jadi lain, codon tu akan membawa maksud amino acid yang lain. And what happen is it will cause major change in protein. Okay, for example, this one. This is missense. Saya padam dulu. missense. Kita ada UAU. Tapi A ni kita gantikan dengan G. Daripada tyrosine, UGU maksud dia adalah cysteine. So dia dah tukar amino acid. Amino acid yang ni dengan amino acid yang ni dah tukar. Okay, kita dah tukar. Dah lain. Okay, dah lain. Kita dah undergo, tadi dah undergo base substitution. Okay, dia tadi dah undergo base substitution. So what happen is, okay, dia punya, siapa yang baru masuk? Okay, dia punya amino acid pun akan bertukar. Jadi amino acid yang lain. Okay, yang tu lah. Okay, jadi amino acid yang lain. Yang tu, cysteine. Alright, itu adalah missense. Okay, missense. Dia membawa maksud amino acid yang lain. So, apa yang awak kena tahu, effect of missense adalah sickle cell anemia. Okay, sickle cell anemia ni awak kena tahu detail. Soalan pernah tanya 3 markah. Explain. Okay, explain the effect of sickle cell anemia, how it happens. Okay, how it happens. Alright. Last one is nonsense. Nonsense mutation. Change one codon into stop codon. Codon tu bila kita tukar, bila kita dah ubah dia punya, kita dah ganti base tu, dia akan jadi masuk codon yang lain. Codon yang lain tu adalah stop codon. So what happen is, it cause the translation to be terminated prematurely. Okay, dia akan cause the termination to be terminated prematurely and the resulting polypeptide will be shorter and leads to non-functional protein. Contohnya nonsense mutation. Yang ni lah nonsense mutation yang ni. Ni adalah nonsense mutation yang tengah tu. Sekejap yang ni. So what happen is we have UAU, but U here we replace with G. So when U is replaced with G, UAG is one of the stop codon. So translation will stop here. Okay, so what happened, we only have this kind of polypeptide chain. So memang tak function lah. Memang tak function. Okay, so ada 3 effect based substitution: silence, nonsense, missense. Untuk missense, you have to know about sickle cell anemia. This one, sickle cell anemia. So sickle cell anemia, on DNA, base T in codon 3 prime, CTC 5 prime is replaced with A. So dalam sickle cell anemia ni macam mana dia boleh terjadi? T ni, kita gantikan dengan A dekat DNA dekat DNA, bukan dekat mRNA. On DNA, the base T is replaced with A. So from 3 prime CTC 5 prime, it will become CAC 3 prime 5 prime. 3 prime CAC 5 prime. So what happen is, the codon will be 5 prime GAG 3 prime in mRNA is replaced with 5 GUG. So sepatutnya codon CTC, sepatutnya DNA CTC dia akan memberi masuk codon GAG. Tapi disebabkan dah berlakunya base substitution, the base T is replaced with base A, CAC akan membawa maksud codon GUG. Okay, so what happen is, the codon is supposed to code for glutamic acid. Codon GAG ni patutnya dia code for glutamic acid. Okay, but this glutamic acid is replaced with valine. Contohnya kita tengok yang ni, gambar yang ni. Wild type maksudnya before mutation. Sickle cell after mutation lah. So wild type beta globin, okay, kita tengok CTC. This T is replaced with A. Okay, T is replaced with A. So what happen is, on the mRNA, it's supposed to be GAG, but it will translate into, it will transcribe into 5 prime GUG 3 prime. Okay, and what happened is, the glutamic acid, the normal hemoglobin, which code for the normal GAG, which code for glutamic acid, is replaced with valine. Okay, so apa yang terjadi, apa yang efek dia, dia akan menghasilkan sickle red blood cell. Okay, dia akan hasilkan sickle red blood cell. Macam ni lah awak kena explain bila soalan tanya. Okay, 3 point yang penting atas tu. Dekat DNA lagi awak dah kena explain. Okay, kalau apa-apa pun soalan tanya untuk gene mutation dekat DNA tu awak dah kena explain. What happened during DNA? What happened to the mRNA? What happened to the amino acids? So sebab awak belajar kan central dogma. DNA kepada mRNA, mRNA kepada polypeptide chain, for this case amino acid. So start cerita daripada DNA sampailah amino acid, 3 markah ataupun 2 markah. Okay? Alright. So the effect is hemoglobin will become abnormal. Okay, sickle. Okay, sickle. So bentuk sickle ni, it will not be efficient. Okay, it will not be efficient to transport oxygen. Okay, sepatutnya dia efficient lah untuk transport oxygen. Tapi bentuk dia sickle, dia jadi inefficient to transport oxygen. Okay, so what happen is erythrocyte becomes sickle shape, it will clump together. Okay, it will clump together and clog the capillaries. This one. Okay, this is sickle cell. Okay, and less oxygen is transported. Okay, so what happened as a result, it will leading to other symptoms throughout the body, including physical weakness, pain, organ damage, and even stroke and paralysis. Alright, so that is the symptoms. Okay, ada soalan setakat ni? Base substitution? Soalan ke? Ada soalan tak? Kalau soalan boleh angkat tangan ataupun write dekat chat. Ada ke? Ada. Okay, kalau tak ada, saya stop sampai sini saja. Okay, saya stop sampai sini. Sebab dah. Alright. Okay, tak ada, saya stop sampai sini saja. Tak ada tak apa-apa. Okay. Alright, so saya jumpa awak pada minggu hadapan and ada lagi sejam, 2 sampai 3, awak boleh pergi semayang dulu ataupun awak pergi lunch. Tapi kalau ada masa yang lebih, bolehlah buat. Okay, saya tengok kerja-kerja yang saya bagi tu nama-nama yang sama juga yang tak buat-buat. Okay, ada juga quiz topik 5 pun, ada juga yang tak buat. So tolonglah buat mana kerja-kerja yang awak tak buat lagi tu, tolonglah buat. Okay, itu saja. Alright, jumpa pada minggu hadapan. Thank you.